Hello there, students from Beehive School. This is teacher Krishna, and welcome to our biology class for week number 13. It's a pleasure to have you all here with me today. And let me tell you that I appreciate a lot the effort that you're doing at home by completing all of the assignments and entering the online lessons that we have prepared for you. So keep it up, and I'm pretty sure that with a little bit of more time, you're going to be doing even better in the lessons here. All right, so before we start, let's continue with some general instructions. Please log into Agilex, enter the Google Classroom, download the Word document, and name it in the following way. The only difference from this document is that you need to add week number 13 at the beginning, so you can keep track of the order of the lessons that you have received in biology, okay? So as we move, remember to take notes on the Word document. That's something very important, because that is going to help you out to keep track of the most important things from here. And it's always very important to start with our end in mind. So for this week, in which we are going to have the module exam for module number 2, our end in mind is the following. Students will be able to review the topics for module 2 in order to successfully complete the module exam. So I have prepared a summary of most of the content from the topics of Module 2 that will help you out to refresh your mind a little bit from what we have studied already. But I recommend that besides the summary that I have prepared for you, you read all of the topics that we have studied, so you can have even more present the information from each one of them. Besides that, I didn't summarize the last topics because I want you to read them and reread them, so you can have a better idea of what you're going to be answering on the exam. Okay, so we're going to start with the topics of properties of water. So we're going to have that, and they have a lot of ionic and covalent bonds thanks to hydrogen. So remember that ionic bonds are like the combination of two different substances by a hydrogen bond, okay? But if we talk about a covalent bond, then we're going to find that there are multiple connections among the elements. And in the case of water, it will be thanks to hydrogen. So one more time, ionic is just one, and covalent are many. Okay. We also have the properties of cohesion and adhesion. Remember, cohesion is how much water can attract itself, and adhesion is how much water can attract other substances. We also have surface tension and capillary action. Surface tension, remember, is the amount of force that you need to break through the surface of a certain element. And we discussed that depending on what state we can find water in, the surface tension will change. For example, the surface tension in solid state of water will be higher than in a liquid state of water. Capillary action, remember, that is the behavior of the substance among a tooth. So depending on their behavior, that is the description of the capillary action of that specific substance. And just to refresh your mind a little bit, we mentioned some examples in class, like how will the capillary action of water will be affected if you add, for example, sugar. If you add a little bit of sugar or if you add a lot of sugar, how would it change? So you can refresh your mind a little bit on what we have discussed. We have that water is a universal solvent. So it is a universal solvent because it can dissolve most of the substances that we can find around. And we also find that it has a neutral pH. So when we talk about the pH scale, remember that the neutral pH for water is 7. And if it goes above 7, then it's going to become a base. And if it goes below 7, it's going to be an acid. Remember, we already had discussions about it in class. We even brought food with different pH like banana, coconut, and several stuff. Okay. Now we're going to talk about microscopes. So Anthony Leeuwenhoek in the 1600s invented the first microscope. So this is definitely a name that you need to remember, Anthony Leeuwenhoek, because with his invention of the first microscope, he was able to observe the first cells, which then helped us out to start theorizing about that all living organisms are made up by cells. Okay. Now, in 1655, the first compound light microscope was created. So that was an invention that supported Anthony Leeuwenhoek as well. Thanks to his investigations, then he kept on experimenting with it, and he created the first compound light microscope. So here we have different types of microscopes. We have the dissecting microscopes. Remember that this one is probably the least powerful one of the ones that we're going to analyze later on. And it has a double ocular lens that you can use to observe a certain sample that you're analyzing. Also, we have a compound light microscope. This one is more powerful than the dissecting microscope. And the compound microscope is like the one we had in class. Remember that you manipulated that you examined some samples from it. You were analyzing paper, you were analyzing hair, and some other stuff. And remember that depending on the lens that you're using, it's going to have a higher amplification. But what is similar in between the dissecting microscope and the compound light microscope is that they both use lenses and light to magnify an image. Lastly, we have our electron microscope. This one is way more powerful than the dissecting microscope and the compound light microscope. And this one does not use light anymore, but it uses like a beam of electrons in order to magnify the images. Okay, we have two other microscopes. The first one is the transmission electron microscope. So this one is pretty similar to the electron microscope. They both have a beam of electrons that surpasses a sample in order to magnify the image. But we have another one that is the scanning electron microscope. So this one is a little bit different because it does not really analyze the insides of the sample, but it only analyzes the surface of the sample that you're analyzing. Okay, and lastly, before we continue, we have an image down there of a microscope. So what kind of microscope is that? So I'll give you just a couple more seconds for you to guess. And that is the compound light microscope, pretty similar to the ones that we have at school. Okay, let's continue. Now we're going to talk about early cells. So, remember that all living things are made up with cells. If we talk about a lion, a monkey, a zebra, a plant, absolutely everything that is living is made up by cells. And we have two different types of cells. We have prokaryotic and eukaryotic cells. Do you remember what is the main difference between both of them? That's something we analyzed as well. Remember that prokaryotic cells are cells that do not have a defined nucleus, such as the one that we can find in an amoeba. But eukaryotic cells do have a defined nucleus, just like the one we can find in plant cells or animal cells. And also we have another important term to analyze, which is homeostasis. So pretty much homeostasis is the regulation of the functions and also the state of an organism. So part of this is, for example, the regulation in your temperature, the regulation in the amount of food that you have to consume in order to have your body working appropriately. That all is part of homeostasis. And also we have some other terms to analyze, which are selective permeability, endocitosis, and exocitosis, as well as passive and active transport. So remember that cells have a membrane. that are surrounding basically all of the organelles. So when we talk about selective permeability, we can mention that that is part of the functions that the membrane has in the cell, because it is going to select only some components that are going to enter the cell when they are necessary. And that's why we have part of the selective permeability. We have endocytosis and exocytosis. So endocytosis is the process in which the membrane of the cell engulfs a certain particle and it lets it come inside the cell. And pretty much that's what happens when a cell needs a certain material for feeding or supplying the necessities that it has. On the other hand, when we talk about exocytosis, it happens when the cell wants to expose material from its body. So pretty much it gathers all of the waste and all of the things that it doesn't need, and then it expels them through the membrane. So that is part of the selective permeability. Also, we have the passive and active transport. So both transports are pretty simple to understand. You have to notice that passive transport is a type of transport that does not require energy in order to take place. But active transport, it does require energy to take place. Okay, let's continue with the cell structure and function. So here we're going to discuss some major parts of the cell. So we have the nucleus. Remember, it contains all the genetic information such as DNA, and it is like the brain of the cell. Nucleolus, it contains the RNA, and it is in charge of creating energy and sending RNA information to other parts in the cell in order for they to create energy and food as well. We have the cell membrane. It is basically a permeable membrane that surrounds cell that is in charge of taking material inside or out of the cell, depending on what's necessary. Cytoplasm and cytoskeleton. So the cytoplasm is like a jelly-like substance where all the organelles are floating inside the cell. And the cytoskeleton is like a skeleton that is very difficult to visualize, but that helps maintain the structure inside the cell. We have the Golgi apparatus. The Golgi apparatus is in charge of packing and distributing material around the cell. We have lysosomes. Lysosomes are in charge of breaking down particles that have come inside the cell. We have the mitochondria that is in charge of building up energy for the rest of the cell. We have the chloroplasts that are in charge of the process of photosynthesis. So remember that we analyzed the process of photosynthesis, and all of that process takes place inside the chloroplasts. And also we have the cell wall. Remember, the cell wall and the chloroplasts are independent for the plant cell. Those are parts that you will not find in the animal cell. We have cellular energy. We have ADP and ATP. So in one of the lessons you received with me, I mentioned that there are some particles that are partially charged and there are some others that are fully charged particles. So in this case, ADP is partially charged and ATP is fully charged, okay? And remember that in order to change an ADP to an ATP, you have to add a phosphate group. So when that phosphate group integrates the particle, it becomes ATP. And remember that ATP storages energy, very short term. So if we want to storage energy for longer periods of time, then ATP is going to change in other particles that we know as sugars and fats. We also have to remember something very important, that are autotrophs and heterotrophs. So remember that autotrophs are the organisms that build up their energy through photosynthesis. They don't need to consume other organisms in order to create their energy. But heterotrophs, they do need to consume other organisms in order to build up and create their energy. So if we have an example of autotrophs, it will be plants. An example of heterotrophs will be animals. Okay. Now we're going to go to our evaluation. So now please read all the topics from Module 2 and complete the evaluation. So as I told you before, I already summarized most of the content. But you still have to read a couple more topics to refresh your mind. When you are done and you feel ready, you can take the evaluation, the module evaluation for number 2. Keep in mind that this evaluation is 10 points, so please take your time and study before you take it. So with that being said, I wish you the best of luck in the evaluation today, and I will be seeing you next week. Goodbye, guys.